Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we are discussing about the different properties of the enzyme. And in this context, so far what we have discussed about the development of the field of enzymology. And subsequent to that, we have also discussed about the nomenclature and as well as the classification of the enzymes. And we have also discussed about the structural studies of the enzyme. So we have discussed about the primary structure, secondary structure, tertiary structure, and quaternary structure of the enzyme. And the previous two modules we were discussing about how you can be able to produce the enzyme in the bulk quantities so that you can be able to use them for various type of applications, such as you can use them for understanding the properties of that enzyme, or you can actually be able to use them for industrial applications. So in this context, we have discussed about the isolation of the gene. We have discussed about how you can be able to clone that gene into the vector of your interest and so on. And once you got the clone, and then you are actually going to transform, or you are going to deliver the DNA into the host of your choice. Then subsequent to that, you are actually going to have a screening assays. You have to perform the screening assays so that you can be able to differentiate between the host which got the DNA and which does not get the host DNA. Okay. So that we are going to discuss in today's lecture. So in today's lecture, we are going to discuss about how you can be able to screen the recombinant clone. So now let's talk about the screening. The screening is a very important aspect, and when you want to screen anything, right? When you want to screen, you should actually going to screen a population based on the exclusive properties when we talk about the vector, the vectors are also, or the clone is actually going to give you the exclusive properties, and these exclusive properties can be exploited for screening the recombinant clone. So let's discuss about this in our subsequent slide. So screening of the recombinant clone. So you can imagine that this is a vector, right? And where you might have cloned. Now this vector is actually going to provide you the various exclusive properties so that it cannot be exhibited by the plain vector, but it can be exhibited by the recombinant DNA. So one of the screening criteria is that this particular recombinant clone probably could express some enzyme, and this enzyme is going to catalyze a reaction where it is actually going to convert the substrate into the product, and this product probably could be colored, or it could actually be able to give you some green or red or blue, some color, and this product is actually going to give the color to the cell, and that's how you can say, okay, blue colored cells are transformed, or blue colored cells are actually taken up the DNA of whatever you exogenously added. The second and the most popular method is that you can actually be work with the antibiotic resistance genes, and this antibiotic resistance gene is actually going to provide the survival of the host cell which actually got the DNA. For example, in this particular vector, what you see is it has the ampicillin gene. So this ampicillin resistance gene is actually going to provide the resistance against the ampicillin. And because the plain vector or the plain host will not actually going to survive, because it will not actually going to have the ampicillin resistance, and that is going to be the criteria how you can actually be able to use the antibiotic resistance genes. The third is the phenotype. Okay, so phenotype is where you can actually be able to use that for that when the cells are going to take the DNA, they are actually going to show you some phenotypic changes. So either of these three broader criteria can be used in different screening methods. So the first method what we are going to discuss is the blue-white screening, and blue-white screening is where you are actually going to use an enzyme to convert the substrate into the product, and that's how this product is actually going to give the blue color to the cell. Okay, so it's going to give the blue color to the cell. So it is actually going to use a chromogenic substrate. The use of the chromogenic substrate to detect a particular enzymatic activity is the basis to screen the desired clone. The most popular system to exploit this feature is called as blue-white screening, where a colorless substrate is processed to a colored compound, right? The colorless compound X-gal, or it's also called as 5-bromo-4-chloro-3-endolyl beta-D-galactosidase, is used in this screening method is a substrate for the beta-galactosidase. The enzyme beta-galactosidase is the product of the LAGZ gene of the lag operon. It is a tetrameric protein, and it is an initial N-terminal region like the 11-41 of the protein is important for the activity of the protein. In this system, the host containing lag gene without the initial reagent, whereas the vector contains the alpha peptide to complement the defect to form the active enzyme. As a result, if a vector containing alpha peptide will be transformed into the host containing the remaining lag Z, the two fragments will constitute to form the active enzyme. In addition, the alpha peptide region in the vector contains MCS, and as a result of insertion of the gene fragment, consequently alpha peptide will not be synthesized to give the fully active beta peptide. The enzyme beta galactosidase oxidizes the X-gal to form the 5-bromo-4-chloryl endoxyl and galactose. The endoxyl derivative is oxidized in air to give a blue colored dibromo-dichloro derivative. Hence, the blue colored colonies indicate the presence of an active enzyme and the absence of insert, whereas the colorless colonies indicate presence of an insert. So, this is actually going to be reversal of what we have discussed, right? So, if the enzyme is active, it is going to convert the X-gal into the blue colored compound, but since we are going to clone the gene of our interest into the alpha region of the protein, and once the clone is going, once the gene you are going to insert that, it will not going to complement the remaining portion which is present in the host, and that as a result it will not going to show you the activity. So the cells which will not show you the activity and remain colorless are actually going to be the transform cells. So this is what we have explained here, right? So the beta galactosidase is a protein which is actually going to be expressed for lag operons. So what you have is you have a lag G gene which has a missing 11 to 41 region. Okay, so if you express that, it is actually going to give you a inactive beta galactosidase, whereas this missing region is actually going to be present onto the vector which actually called as lag Z prime. So this lag Z fragment, when it combines with this inactive beta galactosidase, it is actually going to give you the active beta galactosidase, and this active beta galactosidase is going to convert the X-gal, which is a colorless product, into a blue colored product. And what reaction it is catalyzing? It is actually converting this colorless compound into a blue colored 5,5-dibromo-4,4-dichloroindolyl. Okay, so this is a colored compound, blue colored compound. So since you are cloning the gene into this particular lag Z area, right? You are, even if the fragment is being produced, it is also not going to complement the beta galactosidase, and that's why you are going to have the two scenarios. In one scenario one, when only the vector is present, it is actually going to give you the active beta galactosidase, and that's how it is actually going to be able to convert the colorless X-gal into the blue colored product. But if the insert is present, it is actually going to give you the inactive beta galactosidase, and inactive beta galactosidase will not be able to convert the X-gal into the blue colored compound. Okay, and that's why if you see the reactions or if you see the colonies, what will happen is that you're going to get the blue colored colonies, and you are going to get the colorless colonies. Okay, so these colorless colonies are the colonies where you are going to have your recombinant DNA. Okay, because of the simple reason that it is actually going to have the inactivation of LAC-Z. Then the second criteria is the antibiotic sensitivity. Okay, so antibiotics are the drugs which are actually being responsible for inactivation or the killing of the bacteria, and it happens because the antibiotics disrupt some of the functioning of the cellular properties. Okay, so for example, there are antibiotics which are disrupting the translation steps, there are antibiotics which are disrupting the transcriptions, and there are other antibiotics which are disrupting the protein synthesis. So if you add the antibiotic into the media, it will not allow the propagation of the normal bacterial cells, right? Because it is going to disrupt some of these crucial metabolic pathways. But if you have the antibiotic resistance genes, so most of these antibiotic resistance genes are actually going to inactivate the exogenously added antibiotics, and that's how it is actually going to allow the proliferation of the bacterial cell if they will actually going to have the transformed bacteria. So in this case, vector carries a functional selection marker such as the antibiotic resistance genes and to be used to select the clones. The antibiotic resistant gene product has a multiple mechanism to provide the resistance in the host cell. In this approach, a circular plasmid containing antibiotic resistance can be able to replicate into the host cell plated onto antibiotic containing media. In the cloning of a fragment into the plasmid is cut with a restriction enzyme and a fragment is ligated to give circular plasmid with insert. The transformation of the both DNA species, cut plasmid and the circularized clone into the host and plated onto the antibiotic containing solid media. Only circularized clone will give colonies, whereas cut plasmid will not grow as it has lost antibiotic resistant genes. So this is the table what I have given, and you will see that these are the antibiotics like the ampicillin, kanamycin, tetracycline, chloramphenicol, and these are the gene product from the antibiotic resistance genes. So it will actually going to be beta lactamase, neomycin phosphoryltransferase, ribosomal protection proteins, and the chloramphenicol acetyltransferase. So these are the gene products which are going to be responsible for the inactivation of these antibiotics. And what is the mechanism? For example, in the case of beta lactamase, it is actually going to degrade the ampicillin. Similarly, in the neomycin phosphoryltransferase, it is actually going to make the covalent modification of the kanamycin, and as a result, the kanamycin will not be able to do its action. Similarly, we can have the ribosomal protection protein, which is actually going to have the efflux of the tetracycline outside the bacteria. So that's how, since the tetracycline will not be able to enter into the bacteria, it will not be able to interfere with the protein synthesis. Similarly, we can have the CAT genes, and the CAT gene is going to acetylate the chloramphenicol to acetyl chloramphenicol, and that also is going to interfere with the action of the chloramphenicol. So what we are going to do in this is we are going to have the two DNA species. One is, this is the recombinant clone, where you have cloned the DNA into the MCS and it is going to be circularized. Whereas once you have the cut vector, it does not have the it's circular DNA. Okay, so once you transform and if you put it onto the ampicillin containing plate, this bacteria is actually going to grow because it has the ampicillin resistance gene, and that will actually going to degrade the ampicillin. So it will actually allow the bacteria to grow, whereas when you do the transformation of this cut vector, since the cut vector will not be able to replicate, it will actually will not be able to express the ampicillin resistant gene, and as a result it will not be able to go and form the colonies. Then the third approach is the insertional inactivations. Okay, so insertional inactivation, in this approach a foreign DNA is cloned within the coding gene responsible for a phenotype. As a result of insertion, the gene product is not available to modulate the phenotype of the host. This approach is known as insertional inactivation, and it can be used for a suitable genetic system. For example, in an insertional inactivation of the LAC-Z, right? So LAC-Z is a part of the LAC operon and it is responsible for the synthesis of beta-galactosidase. And you know that the XGAL system can be used to detect the insertional inactivation of the LAC-Z gene to screen the cloned fragment. If the gene is inserted into the LAC-Z, the clone will not be able to produce a functional beta-galactosidase. Hence, blue colored colonies indicate the presence of an active enzyme or the absence of insert, whereas the colorless colonies indicate the presence of an insert. So, this is what you have a LAC-Z which is actually going to produce the functional beta-galactosidase, and that is actually going to convert the X-gal into a blue colored compound, and as a result what you are going to see is you are going to see a blue colored colonies. But if you have the BAMH1 site and if you use this BAMH1 site which is there in the LAC-Z, and if you use that and you will insert, then you are actually going to put your insert within the coding sequence of the LAC-Z, and as a result what will happen is that it is actually going to give you the non-functional beta-galactosidase, and if you have the non-functional beta-galactosidase, it will not be able to catalyze this particular reaction, and as a result it is actually going to give you the colorless colonies. So these colorless colonies are the colonies which are actually going to have the recombinant DNA. So it is actually going to say that okay, recombinant DNA is present. So if you are transforming the vector and if you are transforming the vector which contains the recombinant DNA, the colorless colonies are going to say that it is a vector which contains the recombinant DNA. Then we have the insertional inactivation of the antibiotic resistance genes, and that this example I have taken from the vector which is called as PBR322. So in the PBR322 it has the two antibiotic resistant genes. It has the ampicillin resistant genes and it has the tetracycline resistant genes. Okay, so if a gene fragment will be cloned in SCA1, which is a restriction enzyme, it will disrupt the ampicillin resistant gene, and as a result the clone will be ampicillin sensitive and tetracycline resistance. Okay, so whereas the original plasmid will be ampicillin and tetracycline resistance. To select the clone, first the transformed E. coli is plated onto a tetracycline containing media. Subsequently, a replica plate will be made on the ampicillin containing medium to identify the clone growing on the tetracycline media but not on the ampicillin media, which means if you have cloned a fragment into a SCA utilizing the SCA as a restriction site, what will happen is that it is actually going to disrupt the ampicillin resistance. Okay, so it is going to disrupt the beta lactamase gene. So as a result, this particular clone is going to be sensitive for the ampicillin action. Now what we are going to do is we are going to transform this into the bacteria and we are going to get the colonies. Now this transformed bacteria, what you will do is just make a replica plate of this plate and then you grow this first with the tetracycline. Okay, so when you grow them with the tetracycline, both of these clones are actually going to grow. Okay, and then if you put them onto the ampicillin, what will happen is that this ampicillin resistance is actually going to kill some of the bacteria. For example, in this case, if you compare this and that, what you see is this particular bacteria is being not present here. So, and similarly, you can have some more bacterial colonies which are going to be present in the presence of tetracycline, but they will not be present in presence of ampicillin. So, these are the clone which are actually containing the recombinant DNA. Okay, so what you're going to do is you can just go back and take out these clones from the master plate, and that's how you're going to be able to select the transform plates, or you are going to be select the colonies which contains the recombinant DNA. Then we have the third example of insertional inactivation of the CI repressor. So CI repressor is a protein which is responsible for the shuttling of the virus between the lytic phase and the lysogenic phase. So during an infection cycle, the virus undergoes a lytic and the lysogenic stages, and the CI repressor is a protein which is going to function as the shuttling protein. So The lytic cycle phase is responsible for the lysis of the host to release the virus particle, whereas the lysogenic phase allows the replication of the virus without of lysis of. The CI gene encodes for a CI repressor and which is responsible for the formation of the lysogens. Okay. In the presence of the functional CI, the plaque containing unlysed host cells and has a turbid appearance, whereas in the absence of it, it will be clear. This feature can be used to screen the clone to detect the functional CI or the absence of CI. Okay. So if you have a functional CI, it will say that you don't have the recombinant DNA. If you have the non-functional CI, then it will say, okay, recombinant DNA. Okay. So this is what it is shown here, right? So CI is a temperature sensitive repressor. So if you change the temperature, the CI repressor is going to be expressed and it is actually going to shuttle the virus from the lytic to the lysogenic phase. Okay. And if it is in the lysogenic phase, it will not going to allow the formation of the plaques. But when you clone the protein and you clone it into the CI repressor gene, it is actually going to produce a non-functional CI repressor. Okay. And when you present the non-functional CI repressor, it will actually going to shift the protein, shift the cycle towards the lytic phase. Okay. And as a result, it is actually going to form the plaque, which are going to be which will say that it is actually going to be the presence of recombinant DNA. Then the third approach is the complementation of the mutations. So complementation of the mutation, in this approach, a mutant gene can be used to screen the plasmid containing the missing gene, and the transformant will grow only if the gene product from the clone will complement the function. In general, the gene taking part in the metabolic pathway or biosynthetic pathway are routinely used for this purpose. There are three important requirements in this approach. Okay. So what is the complementation of the mutation is that the host is mutated for a crucial gene. Okay. So this host will not grow until this particular gene product. So it is actually missing with this particular gene. So if you supply the gene product, right, if you supply the gene product which is present on the vector, then this is actually going to grow. Okay. So this is called as complementation, that the host is mutated in such a way that it will not grow until you provide the gene product, and that gene product you are actually going to provide by the recombinant DNA. So in this, there are three requirements of this approach. The host strain deficient in a particular gene. If the gene belongs to the biosynthetic pathway, the mutant host in the case are called auxotroph, as host depends on the gene product or the final product of the biosynthetic pathway as a supplement in the media for the growth. So in some cases, this particular gene could be a part of the metabolic pathway, and gene could be actually be responsible for providing the some crucial biosynthetic molecule. So either you provide the gene product, or you can be able to provide that product into the media. Okay. And as a result, it is actually going to give you the growth of this mutant or this mutated host in the presence of this particular product in media. For example, if this gene is responsible for the synthesis of uracil, okay. So if you don't have this gene, the uracil will not be able to synthesize, and this particular host will not be able to grow. But if you provide the uracil into the media, then if you add the uracil into this media, then this media is actually going to supply the nutrient, and that's how this host is actually going to grow. Then a defined media. So you should have a defined media with the missing nutrients, right? Because while you are doing growing this particular host, you actually can use the media which actually contains the uracil, but you should also have a defined media where this particular nutrient is also missing, so that when the nutrient is missing, the host is looking for that particular nutrient, and that nutrient you will get if the vector is going to supply the gene. And then you also require a vector containing gene to supply the gene product to complement. Now let us see how it works. So you can actually be able to do the complementation to the mutation in a positive fit selection, or you can actually be able to do the negative selection. In the positive selection, host strain does not grow on the media lacking a functional gene, but the host transformed with the recombinant clone can be able to supply the gene product required to grow in the media. So that is called as a positive selection. Positive selection means you are supplying the gene product from the recombinant DNA, and that's how the host is actually going to survive, and it will actually grow and it will give you the colony. Negative selection is that when you are actually going to restore the activity of the gene, it will actually going to kill the transform host. So in the negative selection, a chemical compound is added to the media which will be converted into a cytotoxic agent in the presence of the gene product, and as a result it does not allow the growth of the wild type. But the host strain transformed with the recombinant clone has non-functional gene product, and it grow in the presence of compound in the media. For example, in this particular case, we have taken an example where we have taken an example of URA3. So URA3 is a gene which codes for the orotatine 5' monophosphate or OMP decarboxylase, and an active enzyme process this particular compound which is called as 5-fluoroorotic acid to a toxic compound which is called as fluorodeoxyuridine, and generation of this toxic compound kills the cells carrying the functional URA3 genes. So what we have is we have the gene of URA3 which actually provides a enzyme which is called as OMP decarboxylase, and OMP decarboxylase process this particular compound like 5-fluorouric acid to the fluorodeoxyuridine which is a toxic compound, and when this toxic compound is being generated, it will actually going to kill the cells. This means if you have the functional OMP decarboxylase, it is actually going to indicate that there is no recombinant DNA. Similarly, if you have cloned the fragment within this particular gene, then what you have done is you have done the insertional inactivation of this particular gene. Now, if you have done the insertional inactivation or you have produced the non-functional OMP decarboxylase, this non-functional OMP decarboxylase is not going to convert the 5-fluorouric acid to the fluorodeoxyuridine, and as a result it will allow the growth of these cells and the URA3 cells. Okay, so this is actually going to tell you that if you got the colonies, this means the OMP decarboxylase is inactive. Then these are the methods are more popular in the prokaryotic system. Let's talk about now how you can be able to screen the clones into the mammalian system. So a screening of the transfected mammalian cells. First method is the reporter gene assay. Okay, so in the reporter gene assay system, a chimeric construct is produced with the enzyme gene with an enzyme gene which is cloned in front of the promoter of the gene of interest. The gene reporter gene construct contain a eukaryotic promoter and an enzyme for easy readout. The reporter gene construct is transfected into the mammalian cells with a suitable transfection agents, right? Afterwards, the cells are being stimulated with the agent to stimulate the production of transcription factor to bind the promoter and drive the expression of the reporter gene. A suitable substrate is added to measure the activity of the reporter gene. So this is what you have: the promoter, and you also have a gene, a reporter gene which is going to express enzyme, and this enzyme is going to convert the substrate into the product. And this product readout you can be able to study with the help of the several methods, like you can do the fluorescence, you can do the luminescence, you can also be able to do the UV visibles. Okay. So these are the reporter gene construct what you can use for screening the mammalian clones, like the CAT gene, LAGZ, luciferase, 4A and the GFP. And the gene product are CATG, chloramphenicol transferase. Then Laxi beta-galactosidase, Luc is for the luciferase, and 4A is for alkaline phosphatase, and GFP is the green fluorescent protein. And the reaction what you are going to see for catalyzing is that when you have the chloramphenicol acyl transferase, it is going to run like chloramphenicol to acetyl chloramphenicol and so on. So, in a typical reaction, you can actually be able to use like luciferase, for example, reporter gene system. So, luciferase is an enzyme which is present in the abdomen of firefly, Photinus spiralis. The enzyme utilizes the D-luciferin as a substrate to form the oxyluciferin. In the presence of ATP, magnesium luciferin is getting converted into the luciferin adenylate, involving pyrophosphate cleavage and the transfer of AMP into the luciferin, and the luciferin adenylate undergoes oxidative decarboxylation to form the oxyluciferin, and simultaneously there will be emission of light. The reporter gene construct containing luciferase is transfected into mammalian cell. The cells are washed with PBS and lysed with a lysis buffer. Take the lysate into the luminometer cuvette, and luciferin substrate is injected to start the reaction and measured immediately in a luminometer. So, these are the reactions what your luciferase is going to catalyze, and ultimately it is going to produce the light, and this light can be measured with the help of the luminometer. So, what you are going to do is you are going to first take the look expressing vector, you are going to clone your recombinant DNA into this, right, and then you are going to do the transfection. So, once you got the transfection, you are going to have the eukaryotic cell which has the this recombinant DNA which contains the look gene in front of the promoter. And then what you're going to do is you're going to lyse the cells, and that's how you're going to have the cell lysate, and this cell lysate you can put into the 96 cell plate. You can take the black plate, right, and you can take the negative controls, you can take the positive control and so on, and then you can just put it into the luminometer, right, and what luminometer is going to do is it is actually going to give you the signal for the luminescence, and that signal is actually a light which is going to come from the activity of the luciferase. The reporter gene construct containing luciferase is transfected into the mammalian cells. The cells are washed with PBS and lysed with a lysis buffer. Take the lysate into the luminometer cuvette or plate, and you add the luciferin substrate, and it is injected to start the reaction and measured immediately in a luminometer. The second method is you can actually be able to use some fluorescent protein to look at the transfection of the screening of the transfected mammalian cells. So, you can take the chimeric construct with the GFP protein, for example. So, in the live cells, the GFP protein is a good choice as the reporter gene to screen cells containing recombinant protein fluorescently tagged with the GFP at their C or the N terminus. The cells receiving recombinant DNA will give green fluorescence, and it can be visualized with an inverted fluorescence microscope, and it can be analyzed in a flow cytometer to separate the GFP containing cells from the untransfected cell. Flow cytometer analysis the cell based on the shape, size and fluorescence. A non-fluorescent cell is giving separate peak as compared to the fluorescent label cell, and with the help of the flow cytometer, both of these peaks can be collected in a separate tube. Besides GFP, the other protein what you can use is RFP and YFP and CFP. And so, what you are going to do is you are going to transfect the cells, and what you see in the under the inverted microscope that all the cells are showing a green color fluorescence. And if you want be interested to collect these cells, what you can do is you can just put into the flow cytometer, and flow cytometer is actually going to separate the molecules based on the fluorescence. So, these are the control untransfected cells, these are the GFP expressing recombinant DNA containing cells, and that is how you can actually be able to collect these cells in a separate tube, and they will be the recombinant cells or the cells with the recombinant DNA, cells with recombinant DNA, and these cells you can separate out, and that is how you can actually be able to use them for subsequent experiments. So this is what we have discussed so far, how you can be able to screen the compounds, you can get the suitable clones. Now the question is, if you have done the cloning and you got the suitable clone, how you can be able to verify the clone, okay? So you, because that is very important, right? So you can actually be able to verify the clone by several methods, okay? So what we have discussed so far, we have discussed about the screening of the clone, and now what you got is you got the recombinant clone which are present in the LB agar plate. Now the next question is how you can be able to verify the clone, okay? Because the verification of the clone is very important to that, so that you should not misguide, because you know, and verification of the clone can be done with the help of the DNA sequencing. You can actually be able to sequence the clone to know that whether the gene of your interest is fragment is also present, okay? So the confirmation of the cloned DNA can be done by the DNA sequencing. Historically, there are two methods of DNA sequencing with a similar principle of breaking the DNA, either the chemical method or the enzymatic method, into the small fragment, followed by the separation and analyze them on a high resolution electrophoresis gel. So in a typical DNA sequencing, what you're doing is you're taking the DNA sequence, okay, and then you are breaking that into the multiple fragments, and all these fragments, either you are using the chemical method which is called as Maxim Gilbert method, or you are using the enzymatic method which is called as Sanger's method. So either with the help of the chemicals or the enzyme, you are, you know, breaking this DNA sequence into the smaller fragment, and all these fragments are then going to be separated in a high resolution polyacrylamide gels, and then the signal are going to be analyzed for interpreting the sequences. So let's just first talk about the enzymatic method or the Sanger method. So the first method what is being discovered is the dideoxy chain termination or the Sanger's method. This method is originally being developed by the Frederick Sangers in the year of 1977, for which the Frederick Sangers got the Nobel Prize. In this method, a single stranded DNA is used as a template to synthesize the complementary copy with the help of a polymerase in the presence of nucleotide. The polymerization reaction contains a primer and a nucleotide. So you can have the three normal nucleotides and a 2' 3' deoxynucleotide triphosphate. This means it is actually, you are going to take the single stranded DNA and you are going to perform the PCR with the help of the three nucleotides, which are going to be normal, plus one nucleotide which is 2' 3' DD NTPs. So what will happen is, so you're going to run the multiple reactions of the same, for the same DNA. So but you will actually going to change the DDNPPs. In some cases you are going to take the ATP, in some cases you will take the GTP, in some cases you will take the CTP, and in the other case you will take the TTP. Okay, so this means for every DNA sequence you are going to run the 4 reactions, and in 4 reactions you will take either the DDATP plus you will take the all other remaining 3 nucleotides. Okay, so that's how you are going to you know, make the 4 different types of reactions. When the DNA polymerase utilizes dideoxynucleotide as a nucleotide, it gets incorporated in the growing chain, but the chain elongation stops at the DDNPs due to the absence of the 3' hydroxyl group. In the typical sequencing reaction, you are going to run 4 different DTMPs are taken into the 4 separate reaction and analyzed onto a high resolution polyacrylamide gel electrophoresis. The ratio of NTP and DATP is adjusted so that chain termination occur at the each position of the base in the template. Now this is what exactly you are going to do. In the Sanger's protocol, what you are going to do is you are going to first take the DNA sequence, okay, and you are going to have the terminal sequence. Okay, so then you add the primers. Okay, and when you add the primer, okay, and you are going to have the two options, either you go with the Sanger's protocol or you go with the labeling as well as the termination protocol. So in the Sanger's protocol, what you're going to do is you're going to add the DATPs and the clinome fragments, and you're going to label the DNA, and then you are going to divide this DNA into the 4 reactions. In the reaction A, or that will called as A reaction, you are going to add the DDT ATP and the remaining 3 NTPs. In the second reactions, so this is the reaction number 1. In the second reaction, you are going, which is called as T reactions, you are going to add the DD, TTP, and the rest 3 nucleotides, which are normal nucleotides. Then in the reaction number 3, you are going to add the, or which is called as G reaction, which is called as DD, GTP, and the 3 G DNTPs. And in the fourth reactions, you are going to take the C reactions, which is called as DDCTP, plus the 3 remaining NTPs. And after every reaction you add, you are going to put them for chase, which means you allow the DNA to be synthesized. So in the step 1, a primer is added and annealed to the 3' of the DNA template. The radio labeled ATP is being added to label the primers. Then the step 3, the polymerase reaction is divided into the 4 reactions, and in the step 4, DNA synthesis continue until terminated by the incorporation of the specific DD NTPs, either the A, T, G or C. And in the step 5, a chase of polymerization reaction is performed in the presence of high concentration of NTPs to extend all non-terminated sequences into the high molecular weight DNA. This high molecular weight DNA will not enter into the sequencing reaction. In the labeling or the chain termination protocol, this is the labeling and the chain termination protocol. In the step 1, a primer is added and allele to the 3' of the DNA template. Then in the step 2, a limited amount of NTPs are added along with the one of the radio labeled nucleotide to label the DNA throughout the length. Then the step 3, the polymerase reaction is divided into the 4 reactions, just like as we discussed here. And the polymerase reaction continues with the 4 nucleotide, and out of 4, one of them would be the dideoxy NTPs. Synthesis is terminated at the specific DNA NTPs, either A, G, C or T, to give the DNA fragments of the different length. Now when you got the DNA fragments of the different length, right? From, see, for example, from the same DNA you are going to get first big strand, then you are going to get this, then you are going to get this, like this, right? And so all these fragments has to be analyzed onto a high resolution SDS page, and when you analyze them, you are going to get this kind of fragments. Okay, so you are going to divide that into 4 reactions: A reactions, T reaction, G reaction and C reaction. Now imagine that we have started with this particular DNA sequence. So A reaction, you are going to get a fragment here, right? For T reaction, you got the fragment here, okay? And for G reaction, you got the spot here, and for the C reaction, you got the fragment here, okay? Now what you have to do is you have to run in a reverse orientation like this. You have to walk. When you interpret this, you are going to walk like this, okay? And that's how you are going to get first some A, then AT, then ATT, then ATTA. So that is what you are going to do, and that's how you are going to get the complete fragment DNA or the sequence of the DNA what you have started with, okay?
So in a Sanger sequencing method, what you have is you have a DNA, what you have a target DNA, and that you have to sequence. So what we have, if you want to do a DNA sequencing using the Sanger sequencing method, you have the 2 way. Either you can go with the gel filtration chromatography, gel electrophoresis, or you can do the capillary electrophoresis. So the first step is that where you have to take your DNA into the Eppendorf, and then you have to add the primers. These primers you have to add into the 4 reactions. If you remember, we have said that you have to divide the reactions into the 4 reactions. And then you have to add the DNA polymerase into the each reactions. So you have to add the reactions number 1, 1, 2, 3, and 4. And once you added the DNA polymerase into the 4 reactions, then you are going to add the nucleotides. You have to add the all the 4 nucleotides like C, T, A, and G in all the 4 reactions. And in the subsequent step, you are going to add the dideoxynucleotides. So as if you recall, you can have the 4 different reactions: A reactions, T reactions, C reaction, and the G reaction. And in all of these, you have added the dideoxynucleotide. And what is the difference between a normal versus dideoxynucleotides? The difference between a normal DNTP is that it has the 5' uh, phosphate, and you have the hydroxyl group at the 3', whereas in the case of dideoxynucleotide you have the this OH is missing, and because of this OH is missing, it is actually going to do the it will going to stop the synthesis. Let's see how it is actually going to stop the synthesis. So you can imagine that if there is a DNTPs, it actually will going to form a bond by the phosphodiester linkage, and the OH is still there, so that will continue the synthesis. Whereas in the case of the dideoxynucleotides, once the dideoxynucleotide is going to use its phosphate and going to form the phosphodiester linkage, since the OH is missing on this side, it will not going to allow the incoming nucleotide to bind. So that's how it is actually going to stop the synthesis of the DNA synthesis by the DNA polymerase. Now what you have to do is you have to take the 4 reactions and put it into the thermal cyclers, where you have all the reagents. In the thermal cycler, you have the different steps. Like in the first step, you are going to do the denaturations. So in the denaturation step, you are going to increase the temperature of the thermal cycler. And once you increase the temperature of the reactions, the 2 strands of the DNA are going to be detached, and then you are going to add the primers, and the primer will need, and then there will be an extension. But what will happen is, if there will be a dideoxy TPs, then it is actually going to terminate the reactions wherever the dideoxy will find, the enzyme will find a dideoxy. So what you see here is that the termination is happening at every A. So if it is going to find the A, it will going to give you the DNA of different reactions. Same is going to happen for the dideoxy T reactions. So in all the T, wherever you have the T, it is actually going to terminate the reactions. Same is true for the C reactions. So wherever you could find the C, it is actually going to terminate. Like here, it is going to terminate, and so on. So same is going to happen even for the G reactions. That wherever it is find the G, it is actually going to terminate. So for example, in this case, it has find the G at the end. So it is going to terminate at this point. Then it is going to terminate at this point, and that's how you see they are actually going to give you the different reactions. Now what you have to do is you have to take out these reactions from the thermal cyclers, and then you have to resolve these samples onto the jelly electrophoresis. So you have to take all the 4 reactions and load it into the 4 different wells, and you know that the DNA is negatively charged, so it is actually going to resolve onto the gel electrophoresis. So you load the 4th reactions, and then you connect it to the power pack and you turn on. So when you turn on, the DNA is going to run from the negative to positive, because the DNA is negatively charged, because the DNA is having a phosphate backbone, and that actually gives the negative charge to the DNA. So because of the negative charge, it goes towards the positive electrodes in the gel electrophoresis. And you know that the this migration is in inversely proportional to the size of the DNA. So the smaller DNA will run faster and the larger DNA will run slower. Now what you have to do is, once you have resolved the DNA, you have the two ways in which you can be able to visualize this DNA. You have the either utilization of the radio labeled primers, or you can use the labeled DNTPs. That labeling you can do with the radioactivity. So what you can do is you can use the P32 labeled DNA, a labeled basis, and that is actually going to label the DNA when it is actually going through with the synthesis. So irrespective of whether you use the labeled primer or the labeled ATPs, once you got the DNA being resolved onto the agarose, then you have to do is you have to visualize the DNA band with the help of the autoradiography. So what you have to do is you have to take this gel, you have to take the agarose gel and put it into the gel cassette, put the X-ray films, and then you close it and let it be exposed for overnight or 72 hours. During that period, the radioactivity what is present on the gel is going to expose the X-ray film, and that's how you are going to get the bands of the DNA. Now what, as we discussed before, you have to read it in the reverse orientation, which means you have to read it first this sequence, then 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 this sequence, and that's how you are going to get the sequences from each band. And what you have is, this is your DNA sequence. So what you have to do is you have to take this sequence and then these sequences, and that is actually is going to be the DNA sequence what you are going to get from the Sanger sequence. So this is all about the Sanger sequencing method what we have discussed.
Now the second method is the Maxim-Gilbert method. So this method was discovered by the Maxim and Gilbert in 1977, which is based on the chemical modification and the subsequent cleavage. In this method, a 3' or 5' radio labeled DNA is treated with a base specific chemical which randomly cleaves the DNA at their specific target nucleotide. These fragments are analyzed on a high resolution polyacrylamide gel and autoradiogram is developed. The fragment with the terminal radio labeled appear as a band in the gel, which means what it is going to do is it is going to take a DNA fragment, it is going to label on one side with the radioactivity, and then it is actually going to treat with the chemicals. These chemicals are specific, so they will either going to target the A nucleotide, they are either going to target T nucleotide, the G nucleotide or the C nucleotide, and as a result what you are going to get is you are going to get small fragments of this DNA, where the terminal nucleotide, you know that, so on this side you already have a radioactivity, right, but the place where it is actually going to be cleaved is either A or T or G or C. This means here also you are going to run the 4 reactions, the A reactions, T reaction, G reaction and C reaction, and that is how you are going to analyze these reactions, and that is how they will going to give you the pattern of the DNA sequence. So the chemical reactions are performed in 2 steps, the base specific reaction and the cleavage reactions. Okay, so in the base specific reaction, different base specific reagents are used to modify the target nucleotide. Reaction 1, where you are going to use the dimethyl sulfate DMS, and that is going to modify the N7 of the guanine and then open the ring between C8 and C9. So that is called as G reaction. Then you have a reaction 2, which is you are going to add the formic acid, act as a purine nucleotide. So it is actually act on purine nucleotide. So it is actually going to be called as G plus A reaction by attacking on the glycosidic bond. Then reaction 3, it is going, you are going to use the hydrazine, and that is going to break the ring of the pyrimidine. So it is going to be called as T plus C reaction, because it is not specific for the only C or T. It is actually going to attack on the pyrimidine basis. Then in the reaction 4, wherein in the presence of salt, it breaks the ring of the cytosine, and that is called as C reactions. Once you are done with these base specific reactions, then you are going to have the cleavage reaction. So after the base specific reaction, the piperidine is added, which will replace the modified bases and catalyze the cleavage of the phosphodiester bond next to the modified nucleotide. This means at the end what you are going to get, you are going to get a pattern like this, okay, because here also you have added a radioactivity onto the 5' end. So you're going to have the G reaction, G plus A reaction, T plus C reaction, and the C reactions. The fragment in the G lane is read as G, whereas the fragment present in G plus A but absent in G is read as A. Similarly, the fragment in C is read as C, whereas the fragment present in T plus C but absent in C is read as T. To get the DNA sequence, the band with the lowest molecular weight is read followed by the next band in the 4 lane. For example, this means you are going to start from here and you are going to read like this, okay? But there is a issue. G lane, the band is of lowest molecular weight followed by the band in the A lane, which means if you have the G, okay, and if you also have the same band in the G plus A reaction, this means you are not going to read this, okay. This means this is also going to be considered as G reaction. This means from here you are going to read this, and then from here you are going to read this. From here you are going to see all these two bands are of the same level. This means it is going to be read as C. Then from here you are going to read this as G, and so on. So you will actually go with the you will going to go with the from the lower band to higher band, okay. So for the G, between the G and G plus A, you are going to, if you have the bond in the same line, you are going to read them as G. Similarly, if you have the T plus C band and C band, you are going to read them as C, okay. So if you have got the two bands which are present both in the T plus C and C, then you are going to read that as C rather than T, okay. So same is true for the A plus G and G plus G reactions. So this is the way you can actually be able to sequence the cloned DNA, and that's how you can be able to verify the DNA. So, so far what we have discussed about how you can be able to utilize the different types of tools or as well as the features what are present into the vector and recombinant DNA, and those are the which you can use for the screening of the recombinant DNA. And at the end we have also discussed about the DNA sequencing reactions, so that you can be able to sequence the cloned DNA, and that's how you can be able to verify the clone. So with this I would like to conclude my lecture here. Thank you.